Hello everyone, welcome on the platform of Unacademy. Let's crack CBSE Science. So good afternoon to each one of you. Today we are going to have our discussion on the question paper of 2017 discussion. Okay, so let's start. Before we start, I would like to give my introduction that I am Divya Gupta. I have done MSc in microbiology. I have around 3 years of teaching experience and I am completely passionate about teaching. Okay, just wait a second. So बात करते हैं Unacademy की. That is on the platform of Unacademy there are two different subscription option you people are having. One is the iconic subscription and another is the plus subscription. Okay, so the education in both the subscription will be the same. The only difference is the features of both the subscriptions. Okay, so in the iconic subscription, first you are going to have the personal mentor, means you can have direct one-to-one contact interaction with the educator. Okay, live doubt solving sessions will be there for you. Your parents can also get connected to us, and you are going to have all the benefits of the plus. That are the features of the iconic subscription. While in the plus subscription, you are going to have daily live classes as you daily have. Okay, live test and the quizzes will be held for you. This will help you to analyze the performance. Okay, then structural course will be there for you, and this structural course will help you to best prepare for your particular examination. Hello Sanskriti, good afternoon. Then you are having the unlimited access to all the courses, because the courses are available from class 6 to 12 and for all the competitive exams. Okay, so to have the access to all the free live classes of the courses, you have to make use of the code that is DG007. Okay, so Sanskriti, the earlier lecture, in the last lecture we have discussed about the question paper of 2016 discussion. Okay, what we will get, that we have already seen. This is how the live classes looks like. Okay, you are being prepared with the top educators, expertise. Okay, comprehensive syllabus. And yes, my dear students, if you have not yet downloaded the Unacademy application from your Google Play Store, kindly download it as soon as possible. Make your account over there and select the course for which you are looking for. Okay, so for some days you are going to have the free live classes. After that, you have to take the subscription of the Unacademy. Okay, we are also available on the Telegram application. Download the Telegram app, type this URL link which is there in front of you, click on the join button so that you can have all the information regarding all the updates. Okay, so plus का subscription लेना तो select plus, iconic का लेना हो तो select iconic. Okay, so my dear students, as you all know very well that you are having only 6 months of time for your class 12 preparations, right? So for the plus subscription for 6 months, the amount was 10,000, but now it is just costing 9,000. That is, you are going to have 10% discount. Okay, in your Unacademy subscription. For that 10% discount, you have to make use of the code that is DG007. We call it as a referral code. Hello Ria, good afternoon. How are you? In iconic subscription for 6 months, the cost was 17,000, but now it is just 15,300. That is, you are going to have again 10% discount. And my dear students, this code can be used by any of the student who wants to learn with the platform of Unacademy, whether the student is of class 6 to 12 or for any competitive exams. Anyone can make use of this code. Okay, that is DG007. They are definitely going to have 10% discount. Okay, so that is what about the introduction. Now let's start with the question paper discussion. Just wait a second. Yes. Okay, now let's have our discussion start. We are on the question paper of 2017. Okay, section A, that is 1 mark each questions. Ready? So now comes the first question. Name the stage of Plasmodium that gains the entry into the human when bitten by an infected female Anopheles. Okay, तो female Anopheles mosquito से कौन सा Plasmodium का stage that get enter into the human body? It is the sporozoid. Okay, because ये sporozoids these are present in the female Anopheles mouth. Okay, उसके इस mosquito के mouth में present होता है. Next question. List any two characteristics of the pea plant used by the Mendel. Okay, तो हमें कोई भी characteristics, there were the 7 different characteristics. We can write any of the two. Flower color, position, pod shape, color, seed shape, anyone you can write. Question number 3. Mention the role of the restriction enzyme in the recombinant DNA technology. Okay, so what is the role of restriction enzyme? Is to cut the DNA at the particular site. Okay, to cut the DNA at the specific site, or you can write at the particular site. Fine. Now the fourth number of question. Name the disorder caused due to the absence of one of the X chromosome in a human female. Okay, human female के अंदर अगर एक X chromosome absent है, that is human females normally they have 2 X. Okay, and if one X is absent, then the disorder that will be caused will be Turner's syndrome. Okay, now the next question, that is the question number 5. Name the type of asexual reproduction where the parent cell ceases to exist. Okay, तो हमें इस टाइप का asexual reproduction बताना है जिसके अंदर parent cell are living over there. Okay, ceases to exist, that is fission. because fission में क्या होता है, पेरेंट सेल ceases to exist मतलब इसमें पेरेंट सेल अब उनको अवेलेबल नहीं मिलेगा. ओके, यहाँ पेरेंट सेल विल नॉट बी अवेलेबल. ओके, सिर्फ पेरेंट सेल क्या होते हैं, पेरेंट सेल डिवाइड इटसेल्फ इनटू 2 and the 2 different types of organisms are formed. Okay, fission, binary fission, longitudinal fission or multiple fission you can write. Now comes the section B, that is the 2 mark each questions. Question number 6. I don't think this is coming, man. What? Which topic? Which topic are you talking about? Fifth question. Asexual reproduction का चैप्टर आ रहा है, वो आपको it is not coming. It is coming. It's from first chapter. फर्स्ट चैप्टर में सी पार्ट ओमिट हो गया है. ओके, यस 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 यस, आई रिमेंबर. ओके, फाइन. नाउ द 6th number of question. Name and state the effect of drugs that are often medically prescribed that its overuse lead to drug dependence or the drug abuse. So almost all the steroid drugs. Okay, so ये overuse और over dependent, drug abuse, ये सब चीज़ें किस में आती है, steroid drugs में. Okay, the drugs which are used in order to increase the body's energy or to become more and more aggressive. Okay, so इसमें drugs आते हैं, that is morphine, biurets, amphetamines. Okay, LSDs. What does they do? They affect our central nervous system. They act as a painkiller and they also act as a sedative. Okay, that is treat the depression, insomnia or hallucinogenic effects. Then difference between the pericarp and the पेरिस्पर्म. सो पेरिकार्प क्या होता है? वॉल ऑफ द फ्रूट. एंड पेरिस्पर्म persistent residual of the nucellus. तो nucellus होता है उसका बचा कुचा जो पार्ट होता है, that we call it as perisperm. And pericarp is the wall of the fruit, or you can write wall of the ovary also. Okay. Now the next question number 8. Niches is a part of a habitat. Explain with the help of an example. Niches क्या होता है? The place a single habitat may have different kind of organisms in it, but within the habitat every organism has defined range of conditions that it can tolerate. Resources are utilized and play a distinct role. All together comprises to form a niches. ठीक है, niches मतलब क्या होता है? At a single habitat. ठीक है, एक habitat पे बहुत सारे number of organisms रहते हैं. ठीक है, अब जैसे हम हमारी लोकेशन की बात करें तो we are also living the different types of animals like cow, dog, everyone is living with us. बट उन सबकी जो पर्टिकुलर लिविंग कंडीशंस है वो लिविंग कंडीशंस वही मेंटेन रहेगी. ऐसा नहीं होगा कि जैसे ह्यूमन बींग अपने आप को डेवलप कर रहे हैं तो दे कैन ऑल्सो डेवलप. इफ द ह्यूमन बींग इज अडेप्टिंग देमसेल्फ टू एनी अदर सिचुएशन तो वो भी अपने आप को अडेप्ट कर पाएंगे. इट्स नॉट द कंडीशन. ओके, फॉर एग्जाम्पल पॉन्ड इज हैबिटेट ऑफ वेराइटी ऑफ प्लांट्स एंड एनिमल्स, बट it is gambusia fish that is found in the peripheral part which is its niches. Okay, तो जैसे पॉन्ड है उसके अंदर बहुत सारे एनिमल्स रहते हैं, प्लांट्स रहते हैं, बट गैम्बूजिया फिश हमेशा हमें आपको पेरिफेरल एरिया में ही मिलेगी. फाइन. नाउ नेम द टाइप ऑफ द इम्यूनिटी अ बेबी इज born with. How it is different from the one he gets from the mother after the birth. So in the newborn baby there is a innate immunity. Okay, and the immunity that he or she acquires from his mother is the acquired immunity. Because acquired immunity में क्या होता है? The mother is giving her prepared antibodies to the newborn. Okay, the acquired immunity that is the baby acquire from the mother's milk after the birth. It is a passive immunity. It is an example of passive immunity. तो passive immunity किसके लिए है? For the child. And acquired immunity भी किसके लिए? For the child. Because the child does not have to prepare these antibodies and the child is going to have these antibodies in the prepared form. Okay. Now the question number 10. State the role of AUG codon at the 5' and UAG codon at the 3' end of a certain mRNA during translation. So we all know very well that AUG is a start codon and UAG is a stop codon. Okay, that is what their function is. Now a very important question, that is degenerate and universal silent features of the genetic code. Explain. Degenerate, some amino acids are coded by more than one codon. ठीक है, that is बहुत सारे, जैसे आपने जब वो जेनेटिक कोड का टेबल देखा होगा, in that you have seen that the 4 different codons were coded by only single amino acid. That is the degeneration. Okay, एक amino acid, because codons they are around 64. Okay, there are actually around 64 codons are there and 20 only 20 amino acids are there. Okay, तो 64 codons के कंपैरिजन में अगर 20 amino acids हैं, बट ऑब्वियसली देयर शुड बी सम डीजनरेशन, है ना. तो सिर्फ एक मिथियोनिन और ट्रिप्टोफैन 2 अमीनो एसिड ऐसे हैं दैट आर कोडिंग फॉर ओनली वन कोडोन एंड द अदर रेस्ट अमीनो एसिड्स दे आर कोडिंग फॉर द मोर देन 4. ओके, 4 या मोर देन 4. नो, आई एम नॉट एट ऑल फीलिंग स्लीपी. थैंक यू सो मच फॉर योर सजेशन. As I'm not well, that's why you might be feeling that. Now the question number 11. Write the 3 basic facts that are highlighted in Mendel's law of dominance. ठीक है, तो Mendel's law of dominance के हिसाब से what are the 3 basic facts? Like their characters are controlled by the यूनिट कॉल्ड फैक्टर्स, ठीक है, जिसको हम जीन्स बोलते हैं, कि बिकॉज मेंडल ने तो खाना खाया मैम, यस आई एम डन विद माई लंच, व्हाट अबाउट यू पीपल, हैव यू ऑल डन विद योर लंच? दैट्स गुड। तो मेंडल का फर्स्ट क्या था, दैट इज द कैरेक्टर्स आर कंट्रोल्ड बाय द फैक्टर्स, फैक्टर्स अकर इन पेयर, दैट इज जीन अकर्स इन पेयर, एंड dissimilar pairs, one member is dominant and other is recessive। ओके, जब दो अलग अलग पेयर्स अलग अलग फैक्टर्स की हम बात करते हैं तो वन इज द डोमिनेंट एंड अदर इज द रिसेसिव। दिस वर द कैरेक्टरिस्टिक्स और आई कैन से द बेसिक फैक्ट्स, दैट इज फर्स्ट सेकेंड एंड थर्ड पॉइंट बेस्ड ऑन द लॉ ऑफ dominance। Now comes the question number 12। Name the causative agent of amoebiasis and mention its symptoms। Okay, so this same question was asked in जो हमने आज दोपहर वाला सुबह वाला सेशन अटेंड किया था, that is in the 2016 paper, the same question was there। Amoebiasis causative agent is Entamoeba histolytica, symptoms are constipation, abdominal pain, cramps, stools। okay, हाउ डज इट स्प्रेड, ये क्वेश्चन इसके अंदर एडिशनल आ गया है, उसके अंदर दिस क्वेश्चन वाज ऑफ 2 मार्क, यहाँ पे भी ये 2 मार्क में ही है, बट इस 2017 पेपर के अंदर का एडिशनल पार्ट पूछा गया, हाउ डज इट स्प्रेड, तो इट जनरली स्प्रेड बाय द ड्रिंकिंग वाटर, ओके, विच इज कंटामिनेटेड बाय द फीकल मैटर और food material that is contaminated by the fecal matter, anything can be there as a spread source of this Entamoeba histolytica। okay, then comes the question number 13। what is green revolution, mention the steps that lead to it। green revolution means that is the production। okay, यहाँ पे क्या बात हुई थी, dramatic increase in the food production during the mid 1960s is termed as green revolution। मतलब उन दिनों में the production rate that was increased a lot। okay, mention the steps that led to it, various plant breeding techniques, better management practices, okay, agricultural different practices, these were the steps। name the scientists who contributed to the development of semi draft wheat variety, that was the Norman Borlaug। now the question number 14। are hummingbirds and the fishes regulate or conformers, give reason in support of your answer। okay, so what they are, hummingbirds and fishes, they are the regulators। okay, heat loss or gain is the function of the surface area। okay, now I think this question is skipped, ये इस वाले पेपर में इस साल आपके नहीं है। okay, now the question number 15। GM plants, that is genetically modified plants, are useful in many ways। how would you convince farmers to grow genetically modified plants on their field? Give any 3 reasons। okay, so we all know very well that the genetically modified plants, they are working a lot and they are helping a lot to the farmers, because genetically modified plants है, मतलब उसके अंदर वो सारी desired characteristics है that we require, that is high productive rate, high tolerance rate, okay, then nutritional value internally अच्छा होगा, ठीक है, that is make crops more tolerant to the abiotic stress, because सबसे पहली बात तो ये है कि प्लांट को abiotic stress लो सिट गुड आफ्टरनून। ठीक है, तो सबसे इम्पोर्टेंट बात ये आती है कि प्लांट को एबायोटिक स्ट्रेस से बचा के रखना है, बिकॉज एबायोटिक स्ट्रेस कैन अकर एनी टाइम। ओके, so first is to make the crop tolerant to the abiotic stress. Then it should reduce the chemical pesticides use, help to reduce the post harvest loss. Okay, increases efficiency of mineral usage, enhance the nutritional value. Okay, now the question number 16. Name the male accessory glands in the human and write their function. So there are around 3 to 4 male accessory glands. They are the seminal vesicle, prostate, vulvourethral gland. Okay, they all occur in the pair. Fine. And function: secretion of seminal plasma, which is rich in the fructose and the calcium, which gives high energy to the during the process of lubrication. Okay. Now the next question, question number 17. Write in what hello, good afternoon. Hi. So, write in what contents did the Darwin use the term fitness, survival, selection. Again this question is omitted because evolution वाला chapter अभी हमारे है ही नहीं. Okay, this is the question from the evolution. Okay, and that has been omitted. Fine. Gaurav, hello, good afternoon. Good afternoon. Now comes the next question. Question number 18. State the objective with which a dairy farm is set up. Describe the essential step to be followed for dairy farm management. Okay, तो हमें यहाँ पे दूध की डेयरी खोलनी है. Let's see. So process and the system for increase the yield and improved quality of the milk. Okay, selection of good breeding of the animals. Okay, that is the resistant free, resistant to every disease. Okay, how to have adequate water and kept disease free. Feeding to the feeding in a scientific manner with the quality and quantity to the of fodder. Okay, storage of milk, regulatory inspection of that product and regulatory visit of a veterinary doctor. ठीक है, तो ये सब चीज़ें हमें ध्यान रखनी पड़ती है when we are about to have the dairy farm. Okay. Now comes the question number 19. When Morgan conducted dihybrid cross on Drosophila, like Mendel did with pea plant, the F2 ratio deviated significantly from that of the Mendel's F2 ratio. Explain Morgan and his group gave. Write the explanation that the Morgan and his group gave. Okay. तो यहाँ पे क्या था? Here the case has been combed for the linkage and the segregation. Okay, linkage and recombination वाला case था. That is the Morgan has given. Okay. So when two genes on a dihybrid cross are located on the same chromosome, they do not segregate independently. ठीक है, क्या काम होता है कि अगर दो जीन एक क्रोमोसोम पे दो जीन लोकेटेड है, तो सबसे पहली बात तो ये कि वो कितने एक दूसरे से उनका लिंकेज कितना है. ओके, इज दे आर वेरी नियर टू ईच अदर और दे आर अपार्ट. ओके, इफ दे आर नियर टू ईच अदर तो दे विल सेग्रीगेट व्हेन देयर इज अ क्रॉसिंग ओवर. उसके चांसेस बहुत कम होते हैं कि दो जीन्स अगर पास पास में हैं ओके, तो यहाँ पे क्या हो जाएगा, अगर क्रॉसिंग ओवर होगा तो ज़रूरी थोड़ी कि एग्जैक्टली उन्हीं के बीच में आके क्रॉसिंग ओवर होगा, ठीक है। तो अगर इफ द टू जीन्स आर वेरी क्लोज टू ईच अदर, तो percentage of segregation, that is percentage of recombination, is very less. Okay, and if the two genes are apart, okay, अगर दो जीन्स एक दूसरे से बहुत दूर दूर लोकेटेड हैं, तो देयर आर हाई चांसेस कि उनके अंदर क्रॉसिंग ओवर हो जाए and they can segregate from one another, and at that moment the percentage of recombination can be increased. Okay, so that's why they have got a different ratio as compared to the Mendel's ratio. Okay, और explanation क्या था, their explanation was in terms of the linkage and the recombination. linkage के ऊपर हमने क्या देखा कि how close the genes are located उसके ऊपर है. if the genes are very close to each other, then they will be linked to each other. Okay, and if the genes are very at a very high distance from one another, अगर एक दूसरे से वो बहुत दूर है, at that moment there are high chances of the recombination. Okay, now comes the next question. Okay, draw a diagram of a typical agarose gel electrophoresis. Same question was asked in the 2016. Same question in the different form. मतलब उसके 2016 वाला जो पेपर हमने डिस्कस किया था, उसके अंदर द क्वेश्चन वाज दैट हाउ वुड यू सेपरेट द फ्रैगमेंट्स, हाउ वुड यू सेपरेट द DNA strands. Okay, so the answer was the by helping with the help of the gel electrophoresis. और 2017 के क्वेश्चन पेपर में क्या है, यू हैव टू एक्सप्लेन हाउ टू कंडक्ट द जेल इलेक्ट्रोफोरेसिस. ओके, मतलब आप देख सकते हो क्वेश्चन पेपर डिस्कशन पे ही आप देख सकते हो कि एक साल में क्वेश्चन सिर्फ नाम पे पूछा गया था और नेक्स्ट ईयर में क्वेश्चन के ऊपर पूरा डिटेल्ड आंसर पूछा गया. ओके, तो मतलब कोई पर्टिकुलर क्वेश्चन किस फॉर्म में पूछा जाता है डिपेंड उसके ऊपर करता है. डिपेंड उसके ऊपर नहीं करता कि भाई अगर ये वाला क्वेश्चन पिछले साल आ गया है तो इस साल नहीं आएगा. देखो 2016 में भी आया था, जेल इलेक्ट्रोफोरेसिस 1 मार्क में ही था, अब है ये क्वेश्चन 3 से 4 मार्क में. वी हैव टू एक्सप्लेन दी होल प्रोसेस ऑफ दी जेल इलेक्ट्रोफोरेसिस. ओके, तो दैट्स व्हाई आई ऑलवेज से कि जितना ज़्यादा हो सकता है उतना ज़्यादा क्वेश्चन पेपर आप डिस्कशन देखो. ओके, जितना ज़्यादा हो सकता है आप उतने ही प्रीवियस ईयर के क्वेश्चन पेपर्स को प्रैक्टिस करो ताकि आपको पता चले कि किस फॉर्म में क्वेश्चन पूछा जा सकता है. ओके, अंडरस्टूड माय डियर ग्रेट स्टूडेंट्स. ओके फाइन, लेट्स मूव अहेड. So we have to explain about the gel electrophoresis in the question number 20. And here is the answer. जेल इलेक्ट्रोफोरेसिस में हम क्या करते हैं, एक ग्लास की प्लेट लेते हैं, ठीक है, और उसके अंदर हम अगारोस जेल को फिल कर देते हैं. ठीक है, पूरी अगारोस जेल से ये भर जाती है. फिर यहाँ पे हम कैथोड और यहाँ पे हम एनोड कनेक्ट करते हैं. फाइन, एंड देन हियर ऑन दिस लोकेशंस हम यहाँ पे डीएनए को लोड करते हैं. डीएनए को लोड करने के बाद जैसे हम इलेक्ट्रिक करंट पास करेंगे, तो व्हाट विल हैपन, नाउ दिस डीएनए स्ट्रैंड्स आर दे आर गोइंग टू मूव ऑन दिस साइड. ठीक है, तो आगे आगे मूव करेंगे डीएनए स्ट्रैंड्स एंड दे विल बी सेपरेटेड एज पर देयर फ्रेगमेंट साइज. ठीक है, लेंथ या उसका वेटेज, जितना भारी डीएनए होगा, ओके, हैवियर द डीएनए इट विल बी इट विल ट्रैवल विद लेस डिस्टेंस. मतलब ये वाला डीएनए कैसा होगा, हैवी डीएनए, इसके अंदर ज़्यादा नंबर ऑफ जीन्स होंगे. ओके, न्यूक्लियोटाइड्स. और द डीएनए विच इज़ वेरी लाइट दैट विल ट्रैवल द मोर डिस्टेंस So it will travel up to here. Okay, understood? The cutting of the DNA by the restriction endonuclease results into fragment of DNA. These fragments can be separated by the gel electrophoresis. ठीक है, तो what do we initially do? हम पहले DNA को फ्रेगमेंटेशन कर देते हैं by using the enzyme restriction endonuclease. Okay, this endonuclease it does the fragments of the DNA, and then ये सारे fragments क्या हो जाते हैं? They get separated by the gel electrophoresis. Understood? Now comes the question number 21. Name a terminal method to wait a second. Yes, name a terminal method to prevent pregnancy in the human. Okay, so it is the surgical method or the sterilization. Okay, so the female can have the sterilization or she can undergo the process of surgical method. Name the procedure of a terminal method carried in the human male and the female. Okay, so in the male it is the vasectomy, that is vas deferentia, they are the cut or they are tied. Okay, so that the seminal fluid cannot come out of that testes. Okay, a small part of vas deferentia is removed and tied up to a small incision onto scrotum. Okay, female में होता है tuberctomy, that is fallopian tube is cut and tied up. Okay, these are the very common practices that are done. Okay, so fallopian tube is removed and tied up to a small incision in the abdominal and or through the vagina. Okay, this type of small operation it can be done from the abdomen side or from the vagina also. Okay. Now comes the question number 22. No, it's optional. Question is also there. Do all pollen grains remain viable for the same length of time? Support your answer with 2 examples. No, not at all. क्योंकि हर एक pollen grain की viability period, that is up to which time they can be active, they can be in the active state, वो सबका हर एक pollen grain का अलग अलग होता है. ठीक है, support your answer giving the examples. तो examples के नाम पे हम कुछ भी दे सकते हैं, like cereals, rice, wheat. Okay, so इनके pollen grains they have the viability of around 30 minutes only after their release. तो जैसे ही cereals, rice, wheat के pollen grains release हो जाएंगे, so they can remain viable only for the 30 minutes. It means what? 30 minutes के अंदर they have to come into contact with the stigma of the female flower and they have to get fertilized. Okay, and while there are some plants जिनके pollen grains they are having the viability of more than years. Okay, बहुत सालों तक भी उनकी viability maintain रहती है. Okay, now the B number of question. How are pollen grains stored in the pollen bank? State the purpose of storing the pollen grains in the bank. How they are stored? They are stored in the liquid nitrogen. That is, we call it as cryopreservation. Okay, cryopreservation, that is liquid nitrogen. Cryo means cold. Okay, so in the same way as in our home हमें भी हमारे घर में चीज़ें स्टोर करनी होती है. So where does we store it? Store them for the long period of time in the fridge. Okay, so in the same way, if we have to store the pollen grains for the long period of time, we have to do the process of cryopreservation. Okay, that is we are going to store them in the liquid nitrogen, which is having a temperature of minus 196 degrees Celsius. Okay, why does it required और क्यों preservation चाहिए होता है? For maintaining the viability. Okay, ताकि जो pollen grains है उनकी viability वो active state उनका maintain रहे, preserving the threatened species, because many a times it may happen that the plant is endangered. Okay, plant endangered हो जाएगा, एकदम अचानक खत्म हो जाएगा, so we are going to lose that biodiversity. ठीक है, इसलिए to prevent the threatened species. Then preserving commercially important plants, okay, to be used for crop breeding program. We can also use them for the crop breeding program. Okay. Now comes the question number 22. Expand ELISA. Why is this method preferred over the conventional method of diagnosis of disease? Okay, so ELISA, what is the full form of ELISA? Its full form is that enzyme linked immunosorbent assay. Okay, so ये 4 वर्ड लिखने के you are going to have 1 mark. Okay, so expand the ELISA, that is full form of ELISA. It is asked many a times. Okay, why it is a preferred method? क्यों? बिकॉज एक तो ये बहुत ईजी होता है, इट रिक्वायर्स अ वेरी लेस स्पेस इन ऑर्डर टू कंडक्ट दिस एक्सपेरिमेंट. ओके, और दूसरे बेनिफिट्स देखते हैं. इन्फेक्शन बाय पैथोजन डिटेक्ट बाय द प्रेजेंस ऑफ एंटीजन और एंटीबॉडी अगेंस्ट दैट पैथोजन. ठीक है, तो इसके अंदर हम क्या कर सकते हैं एलाइजा के अंदर, दैट इज सपोज दिस इज अ वेसल. ठीक है, वेसल के ऊपर मैंने ये एंटीजन रख दिया, कि मैंने एंटीजन ऑलरेडी रखा हुआ है. सपोज द एंटीजन इज एक्स. ठीक है, अब मुझे पता है कि शायद अब मैं क्या करूँगी, पेशेंट का ब्लड निकालूंगी और उस ब्लड के एंटीबॉडीज को मैं इसके ऊपर इंसर्ट करवा दूंगी. ठीक है, तो इफ दैट एंटीबॉडीज विल बी प्रेजेंट, दैट एंटीबॉडीज आर गोइंग टू फॉर्म अ लॉक एंड की सिस्टम विद द एंटीजन. ओके, ये एंटीबॉडीज क्या करेगी, इट इज गोइंग टू फॉर्म द लॉक एंड की सिस्टम विद दिस एंटीजन. और जैसे ये वाला सिस्टम बन जाएगा, the disease in the patient can be detected that yes, the patient is suffering from this disease, because patient की बॉडी के अंदर इस डिजीज के अगेंस्ट में एंटीबॉडीज पाए गए. ओके, तो या तो हम एंटीबॉडीज यूज करके हम एंटीजन को डिटेक्शन कर सकते हैं, इट डिपेंड्स अपॉन अस. ओके, तो ELISA टेस्ट इज डन इन 2 ways, whether antigen antibody or antigen or antibody antigen. Okay, conventional method cannot provide early diagnosis which is made possible by the ELISA. Okay, तो क्या होता है इसके अंदर, conventional method cannot provide early diagnosis. दूसरे कोई मेथड हमें इतना जल्दी diagnosis नहीं देंगे as compared to the ELISA, because ELISA, in this ELISA method we are directly using the antigen and the antibody. Okay, और दूसरे कोई टेस्ट के अंदर हम डायरेक्टली एंटीजन एंटीबॉडी चेक नहीं करते. But in this ELISA itself, we are using directly the antigen or the antibody in order to check whether the person is suffering from which disease. Okay. Now comes the section D, that is the from here the HOTS question and the critical thinking questions will start. So my dear students, if, are you understanding everything up to here? So the question number 23. Waste disposal and the waste management poses a major problem in present times. Generation of garbage and its disposal is a major threat or consequently lead to serve the environmental issue. The problem is not the biodegradable and recycled waste. We realize that the need is to reduce the non-biodegradable waste. Why is there a great concern of managing the non-biodegradable waste in comparison to the biodegradable? Explain. तो क्यों हमें ऐसा करना चाहिए कि we have to keep more concern on the non-biodegradable waste as compared to the biodegradable. Okay, so in comparison to the biodegradable waste, non-biodegradable waste pollute the soil and also the underground water, because non-biodegradable जो waste होता है वो degrade नहीं हो पाता है. It remains as it is in the form it was released. ठीक है, जिस form में हम उसको release करेंगे it will remain as it is. Okay, so that's why such type of waste have to be managed very properly, so that उनका release अगर हो भी रहा है तो उनका release इस तरीके से ना हो कि वो soil या water को pollute करे. Such waste stay for a very long time without degradation. ठीक है, तो वो environment में without degradation it remains for the long period of time. और long time मतलब ये नहीं कि 5-6 दिन. Okay, that is 5 million to 6 million years also. Now the B question. As a member of Eco Club of your school, suggest any 2 ways that you will discuss with your fellow member to organize for a zero garbage day once in a month in the school. How you are going to do that? Avoid use of plastic in packing for the milk and the water. Avoid use of plastic in packing for fruits and vegetables. Anyone correct point? Okay, तो इसमें क्या होता है कि we can make the students aware that they don't have to bring anything related to the plastic. Okay, whether they are bringing it with the tiffin box or whether they are bringing with the water bottle, anything. Okay, so one day should be there in which they are not going to bring anything related to the plastic. They have to use only the bags which are made up of clothes. Okay, now the next question. Here comes the section E, that will be of the section E 1, 2, 3, 4. Section E is the 5 mark questions. Now let's start with this question. Yes, they are the 5 mark questions. So we have to identify the first, second and third. Third is the fetus. Okay, second is the umbilical cord, and first is the uterus wall. Identify the plant. What? First is the villi, placental villi. Okay, draw the label diagram of blastocyst. What is parturition and explain and how it is induced at the end of pregnancy in the human female. Okay, so first was the placental villi that were on the top. Okay, then there was a umbilical cord, and then there was a embryo. Okay, now we have to draw the label diagram of human blastocyst. Blastocyst, it is having 2 main parts, that is inner cell mass and trophoblast. Okay, inner cell mass जो in future ectoderm, endoderm और mesoderm बनाएगा. Trophoblast it gets in contact with the uterine wall. ठीक है, trophoblast क्या करता है, uterine wall के साथ merge हो जाता है, and it helps to get the developing embryo come into contact with the female body. ठीक है, मतलब यहाँ पे इसका क्या हो जाएगा, trophoblast क्या होता है, implantation. Trophoblast helps the embryo in the process of implantation. Inner cell mass, in future they are going to grow in the 3 different layers, that is ectoderm, endoderm and mesoderm. Now comes the next part, that is process of delivery of the fetus. Now, a signal of parturition originates from the fully developed fetus. Okay, so signals of parturition, they are originated from the fully developed fetus and the placenta. दोनों के through ही the signals are generated. Okay, and it causes the mid-uterine contraction that we call it as fetal ejection reflux. This triggers the release of the oxytocin, and during that moment in the female body, the oxytocin is released. Okay, what this oxytocin does, it makes the uterine wall to more and more contract, and due to that contraction, stronger and stronger contraction leads to the parturition. Okay, now comes the optional question of this. My dear students, do always remember कि optional question हमेशा same chapter से ही आएगा. Okay, now which labeled diagram depict the stage of the embryo development in a dicotyledons. Okay, endosperm development proceed to the embryo development. Why? That you have to answer. So is this, that is zygote develop होता है globular embryo में, that gets developed into heart shape embryo, and that gets developed into mature embryo. Mature embryo के अंदर radical, plumule and cotyledon, they are very easily available. Endosperm is filled with reserve food material which are used for the nutrition to the developing embryo. Okay, so what does the endosperm do? It is reserving the food material, and that reserved food material will be given to the developing embryo. Now the question number 25. How are polygenic inheritance and multiple allelism is different? Explain with the help of an example. So polygenic inheritance means controlled by 3 or more genes. ठीक है, कोई भी कैरेक्टरिस्टिक की जब हम बात कर रहे हैं, when it is controlled by more than 3 or more than 3 genes, it is called polygenic inheritance. Poly means many genes. Okay, multiple allelism means for a particular characteristic, okay, there are more than 2 alleles. Okay, generally दो तो होते ही हैं, but कुछ characteristics में there are 2 or more than 2 alleles. इसको हम बोलते हैं multiple allelism. एग्जाम्पल अगर हम देखें, जैसे ABC जीन कंट्रोल स्किन कलर. ओके, जैसे ह्यूमन स्किन कलर है, ठीक है, तो स्किन कलर आर नॉट ओनली 2 इन द होल वर्ल्ड. स्किन कलर, देयर आर मेनी डिफरेंट टाइप्स ऑफ स्किन कलर, and there are many different types of combinations also. Okay, and multiple allelism में क्या है, that is blood group is the best example for the multiple allelism. Okay, now list the criteria a chemical molecule must fulfill to be able to act as a genetic material. तो कोई भी chemical molecule को अगर it has to act as a genetic material, what is the criteria? तो जेनेटिक मटेरियल के पास कैरेक्टरिस्टिक्स कौन कौन सी थी, जब आपको पता है, देन यू कैन राइट वेरी इजीली द आंसर. इट शुड बी एबल टू रेप्लिकेट, दैट इज उसको अपने रेप्लिका, उसको अपनी कॉपीज बनाने आनी चाहिए. इट शुड बी केमिकली स्टेबल. ओके, बिकॉज अगर वो केमिकली अनस्टेबल होगा then how it can act as a genetic material? It should provide a scope for the slow change and mutations that is required for the evolution. तो ऐसा भी नहीं होना चाहिए, एकदम रिजिड भी नहीं होना चाहिए कि भाई उसकी वजह से म्यूटेशन ही नहीं हो रहे. स्लो एंड ग्रेजुअल म्यूटेशंस आर रिक्वायर्ड इन ऑर्डर टू do the process of evolution, okay, as per the latest conditions. It should be able to express itself in the form of Mendelian character. और उसके पास Mendelian characters को मतलब जिस टाइप से Mendelian characters के हमने पढ़े थे, law of dominance, segregation, independent assortment, ये सारे characteristics भी that उस chemical के पास होनी चाहिए if that chemical has to work as a gene. If that chemical has to work as a genetic material. Now comes this experiment, and yes, this experiment is skipped. Now comes the question number 26. What does the ecological pyramid indicate? Explain the three different types of the upright pyramid in nature with the help of an example. Okay, ecological pyramid क्या बताता है? It show us the relation between the different organisms. ठीक है, अलग अलग लेवल में अलग अलग ऑर्गेनिज्म का रिलेशनशिप क्या है एक दूसरे से, that is being indicated by the ecological pyramid. Okay, अलग अलग टाइप की पिरामिड कैसे होते हैं? Pyramid of number, that is example is a grassland. That is in the grassland ecosystem the herbivores are more as compared to the carnivores. Okay, pyramid of biomass, that is according to the number, that is biomass, sorry, कि कितना वो मटेरियल प्रोड्यूस कर रहे हैं. फॉरेस्ट और ट्री इकोसिस्टम, producers have more biomass than herbivores and carnivores, but obviously जो producers होंगे they are producing more biomass as compared to the herbivores and the carnivores. Pyramid of biomass shows a sharp decrease in the biomass in higher trophic level. तो क्या होता है, बायोमास के कंपैरिजन में producers they are having the highest biomass, then the level of biomass gets decrease, and at the highest level the biomass shows sharp decrease. Okay, now pyramid of energy, that is grassland ecosystem. Producers have more energy than the herbivores and carnivores because they are using directly the sunlight. Okay, producers they have more energy as compared to the herbivores and the carnivores. And एनर्जी लेवल क्या हो जाता है, सिर्फ 10% आने वाले नेक्स्ट टॉपिक लेवल पे पास होता है. ये तो आपने पढ़ा ही होगा. दैट इज अगर प्लांट्स 100% एनर्जी कंज्यूम कर रहे हैं तो उस 100% एनर्जी में से सिर्फ 10% एनर्जी हर्बीवोर्स को जाएगी. हर्बीवोरस से सिर्फ 10% एनर्जी आगे जाएगी, मतलब कितनी कार्निवर्स के पास आई, ओनली 1%. नाउ द नेक्स्ट क्वेश्चन. Indiscriminate human activities such as alien species invasion, fragmentation and habitat loss have accelerated the loss of the biodiversity. Justify by taking the one example from each. Okay. So in this question what we have to do? वही बताना है biodiversity loss के बारे में. The main reason. Alien species invasion. When alien species are introduced, alien species means the unknown species और दी स्पीशीज दैट हैव नेवर लिव्ड इन दैट पर्टिकुलर एरिया. अगर उस एरिया में एक नई टाइप की स्पीशीज को लाके रख दूंगी तो इट इज गोइंग टू कॉज अ वेरी ग्रेट डेंजर टू दी स्पीशीज दैट आर ऑलरेडी लिविंग ओवर देयर. ओके, दैट इज अफ्रीकन कैटफिश को डाल कर दिया पॉन्ड में, रिवर्स में, ठीक है। एंड देयर आर मेनी अदर एग्जाम्पल्स। वेट अ सेकेंड स्टूडेंट्स। ओके फाइन। Justify by taking one example of each. Alien species invasion हो गया. Another question is state the importance of IUC and red data list, hotspot in conservation of biodiversity. Okay, so red data. Yes, habitat loss. Amazon rainforest is being cut and cleared by cultivating the soya beans and conservation of grassland for raising the cattle. ठीक है, तो ये चीज़ें जो हो रही है बहुत कॉमनली हो रही है इन द एनवायरमेंट, दैट इज द पीपल आर कटिंग द बिग फॉरेस्ट एंड इवन द ग्रास लैंड ऑल्सो, ठीक है। उसकी वजह से क्या हो रहा है, द एनिमल्स एंड द स्पीशीज दोज हु आर लिविंग इन दैट फॉरेस्ट, दे आर नॉट हैविंग पर्टिकुलर एरिया, और उसकी वजह से क्या हो जा रहा है, देयर हैज़ बीन द एक्सटिंक्शन ऑफ द स्पीशीज। रेड डेटा बुक, व्हाट इन्फॉर्मेशन इट इज प्रोवाइडिंग? रेड डेटा बुक इज प्रोवाइडिंग अस द इन्फॉर्मेशन रिगार्डिंग द एक्सटिंक्शन ऑफ स्पीशीज। जितनी भी एक्सटिंक्शन स्पीशीज एक्सटिंक्ट होने वाली है, ठीक है, उन सबकी इंफॉर्मेशन आपको एक रेड डेटा बुक में मिल जाएगी। ओके, रीजन विथ वेरी हाई लेवल ऑफ स्पीशीज रिचनेस, हाई डिग्री ऑफ एंडेमिज्म, स्पीशीज confined in that region are not found anywhere else, are identified as need to be conserved in the priority basis, ठीक है। काफ़ी सारे ऐसे ऑर्गेनिजम्स होते हैं, ऐसे स्पीशीज होते हैं जो सिर्फ आपको एक पर्टिकुलर एरिया में ही मिलेंगे, उस पर्टिकुलर लोकेशन पे ही मिलेंगे, एंड इवन दिस टाइप ऑफ ऑर्गेनिज्म कैन नॉट सर्वाइव इन एनी अदर प्लेस ऑल्सो। सो उस टाइप की जगहों को, उस टाइप की प्लेसेस को प्रिजर्व करना बहुत ज़रूरी है एट द प्रायोरिटी बेसिस, बिकॉज if they will not be preserved, the organism will not be able to live in the any other area also, ठीक है। So that is what about the today's session, that is 2017 का क्वेश्चन पेपर डिस्कशन था ये, ठीक है। So thank you my dear students for attending my today's lecture. I hope you have liked my lecture. Don't forget to make use of the code, that is DG007, so that you people are going to have 10% discount in your Unacademy subscription features. Okay, so we'll meet again at 9 PM today on the platform of Unacademy with some new interesting topics. Kindly join me on the platform of Unacademy at 9 PM tonight. The link in the description box, everything is there related to my information. Okay, till then, bye bye everyone. Thank you.